All right, guys, so let's look at mutations. So what happens when there is a change in the DNA? And so the original, remember, we go from DNA to RNA to protein. So if we change the DNA, what's going to happen? So let's look at our learning objectives. I'm going to make myself small. Okay. All right, so how does that work? How do mutations affect the gene? And right, and how do mutations affect genes? So mutations are hereditable changes in genetic information. So hereditable means that they have to ultimately affect the germline or the reproductive cells in an organism. All right, so that change is passed on to the next generation. If you have mutations in existing cells, that change has no effect on a population or a species in general because it's not passed on. Okay, so you can get mutations that'll cause skin cancer, but you're not going to pass that on to your kids. So we'll look at two basic categories, gene mutations and chromosomal mutations. And so what happens when we actually look at genes? So there's a couple of types of gene mutations that we're going to look at. And one is called a point mutation. And point being at a point. A point mutation is a change in a single nucleotide. And under the category of point mutations, we have three types of mutations. We have a substitution mutation. So originally, this had a C here. So originally it was CCA. The C was substituted by a T, and the T, so now we have GTA. Okay. So the corresponding RNA code that comes off of there will be a CAU. Originally, it would have been a C, and because you had a C here, it would have been a CGU. So that changes the amino acid. Remember, these threes are called codons, groups of three, and each one of those code for an amino acid. And amino acids are what builds the proteins. And so if you change the amino acid, you potentially change the shape of the protein. And because life is three-dimensional, it may or may not have an effect on the organism. We have another type of mutation called an insertion, where a gene is inserted into this this sequence. And what happens then is, if you look at the bottom here, we're looking at these are just three-letter abbreviations: MET, HIS, theremin, ther, and phi, right? So if we do an insertion here, we add a nucleotide here, adenine in this example, then that shifts everything to the right of it down one notch, which shifts the sequence of amino acids after that. Have a major effect on a protein, depending on where that occurred in the gene. Okay, if it's the last two proteins or last two amino acids, it might not have a big effect. If it's in the beginning, it might have a major effect. Okay. So there are many examples of single gene mutations that cause disease. For example, cystic fibrosis is one. Deletion is when an amino acid or a nucleotide is pulled out, and then that shifts the subsequent amino acid. So we lose a nucleotide here, and that shifts them to the left, and then creating a different result there. So let's look at the result of a substitution mutation. So on the top, you're looking at the normal sequence of events. And so we have AUG, which is the start codon, MET or methylene right here. So we got A, U, G, and that's going to code for that amino acid. And then we have A, we have, GCA, the corresponding RNA strand is going to be a CGU, and so the CGU is C, G, and U, arginine. Okay, so that's how you read that. I guarantee you're going to see this again. So then you have a DNA strand, AAA, and that's going to pair with, remember in RNA, it's not T, it's U. So U. So U, phenylalanine. Okay. So if we have a point mutation in there, that changes one. We're going to focus on this C here. So this C is going to be popped out, and a T is going to be put in through a variety of mechanisms that can happen, copying error, exposure to radiation, a whole bunch of different ways that we can have mutations in a cell, in a DNA. And because that thymine is now here, it's going to pair with an A. So it's going to change it from CGU to CAU, which is histidine. So you're going to change the amino acid from arginine to histidine. Okay, so what's the result of that? The result, I'm going to tell you this, I'm going to tell you this many times. The result could be good. The result could be bad. The result could make no difference. Okay. All right, so then if we're looking at further consequences, CCA or CCU are proline. So if we end up changing that to a different one, then we would end up there. Okay, so this is a point mutation substitution. So a substitution is going to have an effect typically on one amino acid. Now, know that it cannot have an effect at all. So if we have another, we have a CC, we have a code that's going to code out for CCC, and we had a substitution that then made a code for CCA, they're still going to code for that proline. So a mechanism that creates no difference is you do have a change, and a change is a mutation. Okay. Now, point mutations. Point mutations can occur in two different ways. An insertion, we can put something in, or a deletion, we can take something out. And each time that's going to result in what we're going to call a frame shift mutation. And so it's going to change the amino acids, possibly change the amino acids from that point onward in the reading process. So if we're over here and we have an original set of DNA here on the top, and then this is the RNA that would be coded, AUG, it's the start codon, CGU, arginine, okay, ACC, and then UUU. So if we have an insertion here, what we're doing is we're actually shifting this part of the code to the right. So to the reading area. So we're going to change this codon from CGU to UCG, which changes the amino acid from ARG to SER. And because then that one shifted, everything that recurs after that is going to change. So these are now different also. So that's a point mutation. Insertion, deletion, pull one out, frame shifts it to the left, and then you end up with, again, you're pulling out a G here instead of inserting one. And so that G is gone. So then we're going to start with a GUA here, GUA, which is codes for valine. And then again, CCU is proline. So these are the codons. You may see a question at some point is how many codons would it take to code for an amino acid with this many, or a protein with this many amino acids? And it's always going to be 3 times that amount. Okay. All right, so those are point mutations. That's a change in the DNA. So sometimes the chromosomes have mutations, and that can happen through a variety of mechanisms. Again, exposure to damaging carcinogenics or damaging things that damage the DNA during cell division will do that as one mechanism. So we can have a deletion where we have an original chromosome, A, B, C, D, E, F. And then here, and we're looking at pairs of chromosomes again. So here we have a pair of chromosomes and the B is gone, C, D, E, F. So this is missing a gene for something. That's what they're denoting, where somehow a gene got deleted. Now, if this chromosome was passed on to the offspring, that would be a problem. Duplication. So sometimes we can have double genes in there. And again, we go back to the result of which is could be something bad, could be nothing at all, could be something good. So it depends. Okay. We could have an inversion mutation where you literally just change the direction that they go instead of going ABC, it's going ACB. So that's inverted. And then a translocation is when you get a region of chromosome from another chromosome into a chromosome it wasn't originally in. So the addition of chromosomal material in here. And so that would result in ABGHC. Okay. So those are the different types of chromosomal mutations, and they can result in depending upon what that region of the chromosome codes for, it can result in a variety of problems, or not problems, or whatever. Okay, so again, as I said, they can harm, help, or have no effect on an organism. Some mutations arise from mutagens, chemical or physical agents in the environment. UV radiation, nice young person has seemingly really healthy skin. You're not worried about it. Well, that's fine. You need a certain amount of sun. The long-term effects and what you should be aware of is that sun damage is cumulative, which means that the damage your skin gets from the sun builds up over time. So every sunburn you have is like pennies in the bank. And eventually you end up with different types of damage in different areas in your body, depending upon how much exposure you have. And again, the skin then is right, is going to take the grunt of that damage. And there's other mutagens that happen, chemical and physical agents. If you're we used to say smoking, but now you guys are all vaping. That's going to be something that it's just going to be a mess. Once again, chemical agents being inhaled into the lung is a really good way to alter the DNA in that area. And so we're going to see a pretty significant rise or a consistent, I should say, probably more than likely just a consistent case rate of lung cancer and other things that are associated with tobacco and usage. Because, and that's just a speculation. You can't say that it isn't bad. For one thing, your lungs didn't evolve to suck that stuff into them, period. So, yeah. All right, so a harmful mutation. So some of the most harmful mutations dramatically change protein structure. So there's a sickle cell anemia, for example, changes a amino acid that makes up your blood, your red blood cells, when red blood cells are made. So when that amino acid is changed, it makes a different amino acid and it puts it in that chain of amino acids. And so when that protein folds, that is making this normal looking blood cell, the protein that's with the different amino acid in it, then has a different shape, and so it interacts with other proteins differently, and the blood cells end up being sickle shaped, literally sickle shaped, and that causes problems. They tend to clump and create a whole bunch of problems with circulation and oxygen delivery to the entire body. Okay, beneficial mutations can produce proteins with new or altered functions that can be useful to organisms in different and changing environments. And so, we have a beneficial mutation within an environment is going to we're going to take that and we're going to stack that on top of population growth. And so then you have a mutation that occurs in a population that gives it a benefit in that environment. And so if an animal has an enzyme that lets it that digests a certain plant better than other animals, then that animal has a competitive advantage in being able to digest and get energy. And then the offspring of that animal, again, we can spread that trait across a population. That population would have a competitive advantage in an environment like that. And mutations occur throughout. We'll be looking at mutations and their effects on population in terms of biological change over time, aka evolution. And so how random mutations then can give a competitive advantage to an organism within a population, then a population gets to grow. Yeah. All right. That is mutations in a nutshell. Certainly lots more to learn, but we'll stop there.